Well, thank you very much for inviting me today. It's kind of a strange time, and it's the first time I'm having an online talk, so bear with me if I'm losing the track. So today I'm going to speak about the work we've been doing for some years now on Barley recombination and Barley meiosis. And you're right, I'm a cytologist, so there will be a lot of pictures, but hopefully you will get the message what I'm trying to share at the end. So if you look at the species in the front of this picture, the humans, we have like a relatively large genome already. It's 3.4 gigabyte, and then it's organized in 23 chromosomes, and it's a bit special organization because we have 20 autosomes and then 6 chromosomes. If you look at the Barley genome, it's almost twice as big as our genome, but it's actually only organized in 7 chromosomes. So chromosomes are a little bit more larger. And we have a lot of repetitive DNA in here as well. If you look at those pictures on the bottom of the slide, this is from a paper from the Lindsay et al. And if you look at the scale bar, which is about 10 micrometers, it's roughly about 20 micrometers diameter for the leptotin zygotenamal myocyte. Now, if you look at the Barley one, if you take the same scales at 10 micrometers for a similar stage of meiosis, it's actually a little bit smaller. The level of compaction is a bit higher. Now, if you move to the model plant, Arabidopsis, the first thing you can appreciate is you can hold it in your hand as compared to the massive barley plant that actually behind the humans in the front picture. And it's got a very tiny genome. It's got only this 135 MB. And is organized in 5 chromosomes. But it's been brilliant because you can sequence it, you can do a lot of things, and it has been very valuable for the meiosis field in particular. So if you're using, for example, high super resolution microscopy, you can have quite lovely pictures and things. This is one from the paper from Lambing et al., which quite recent actually. But if you look at the scales, it's actually very small nucleus in here compared to Barley. And you can carry on like this by moving to a much larger crop here, like wheat, for example. So wheat has a more complicated story here because not only the genome is 3 times larger than the Barley one, but it's got also, it's got a polyploid. It's got 3 different genomes at once, and it's organized in 21 chromosomes. That makes 42. Now, if you look again, one of the pictures we've generated with collaborators in France, we can see that the 20 micrometers is roughly the size of the myocytes. And it means that the genome is even more compacted here. So the chromatin organization in those large genome spaces is very important, and it's quite a very important thing into meiosis. Now, if you look at Barley and the recombination in Barley, you can actually divide the Barley chromosome in 3 different zones. So this is something that has been done in this paper from Marcher et al. In 2017. And you have basically 3 rough regions. So the first region in the dark blue here is at each end of the telomeres and is the end of the chromosomes. It's gene rich, and this is where most of the recombination happen. So if you go into the middle section here, under which is on either side of the centromere, you actually have medium recombination. And in the centromere, which is the region in the middle, you have very low to absolutely no recombination in some extents. And the problem is, it doesn't mean that there's no gene in here. It's actually about 20–30% of the gene that actually really rarely recombine and are excluded from the breeding process. And it's very bad for us, especially with all this problem with the climate change, because there are actually quite a lot of interesting genes, such as pest-resistant disease and stuff like this. So you really, really want to accelerate breeding to actually unlock recombination in Barley. And if you want to know more about the project that we've been working on so far, I invite you to click on this link that leads you to the YouTube video of a Shuffle project. So where you find meiosis in Barley? So meiosis in Barley is here. So this is where my fingers are. So I appreciate the nail polish here. This is a 6–7 weeks old plant. So it's quite nice. It's not very tall at this stage. And you really need to feel what I call the spikes in it. So if you open this stem, this is where the spike is located. So the spike we're working on, this is one of the largest spikes that we're working on, is about 18 millimeter. And to give you an indication of the size, 19 millimeter diameter is the diameter of a 1 penny coin. So it's actually not that big. We are working as well with smaller spikes if you want to have early stage of meiosis. And within each of the flower here, you have like 3 anthers that will be synchronized. So if you go further down, so if you cut these anthers in half, this is what you obtain. You have each locus here contain meiocyte, and if you cut transversally, you actually can see and appreciate the synchronicity of the meiocyte within the tissue. And meiocyte is about 8%, 10% of the number of cells in one anthers. So meiosis is a very specialized cell divisions. It's aimed to produce gametes, and in order to produce gametes, it starts with diploid cells, and it will end up with 4 haploid gametes. And each of the gametes have a very specific allelic combinations. So in Barley, it starts with something called the telomere bouquet and telomere cluster. So all the telomeres would cluster in one side of the nucleus, and it will help to promote homologous search, but it's not the only way to do this. So using immunocytology and using specific protein against meiotic protein, for example, ACE-Y1, which is an axis protein, which will label the chromosome in green, and ZIP1, which is the central element protein that will label the synaptonemal complex, which I will detail a little bit better. You can actually follow and track meiosis. You can also track meiosis by just doing a DAPI staining and DNA in situ hybridization. So here's an example of a packet in cells where you see that 45S rDNA probe and 5S rDNA probe are paired up. So the process of homologous pairing is complete. Diplotin, you can already appreciate that the level of compaction of chromosome is actually high. You see here the chromosome is very elongated, but here they're really compacted because they need to move to the next stage and separate properly. Diakinesis is an interesting stage because you can already start to visualize the chiasmata here. I labeled one in an interstitial one here with the little stars. And at metaphase, you can actually see them much more better. Here's a very distal one, here an interstitial one, and here another distal one. And in Barley, you have what we call 7 bivalent, or the ring donut shape bivalent, and is very specific to the bivalent formation in Barley. And the second stage, the second division of meiosis resembles a mitosis, although there's a lot of little differences now that we know. But the first step is to separate the homologs. So these two, for example, if you take this bivalent here, will separate, and one chromosome will go to one side, the other one will go to the other side. And the second phase will separate the chromatids. And this is how you end up with each gamete and then containing an allelic diversity, which is very unique. Now, I told you I was going to explain a bit more the synaptonemal complex formation. So if you look at the first stage of meiosis here in what we call zeptatin zygote, you can see that all the chromosomes now have the axis formed, which means they are prepared for chromosome synapsis. And you can appreciate that here there's a fork where the 2 homologs are not paired up, and they start to be paired up by the magenta protein called ZIP1. Now, if I move mid-zygote, you can see that the chromosome is halfway paired. So again, you have 2 homologs aligned along each other with the ZIP1 in the middle, and this one is completely unpaired yet. Now, if you move to packaging, the process of synapsis is complete. And not only is complete, and then the chromosomes start to twist around each other, so we can compact further for the next step. And in diplotin, this is where the synaptonemal complex will start to dissolve, and we start to see what we call, well, what I call the tinsel chromosomes. The little story behind was that I found it near Christmas time, so this is why I call tinsels. But you can appreciate that the way the genome is organized, again, is completely different than in other organisms. So just a small break on the synaptonemal complex dissolution, because I promised to explain this a little bit. So if you look at the way the ASY1 protein is getting out of the axis, it's actually quite interesting because first of all, ZIP1 will dissolve, but some of the loops of ASY1 loops actually form before. And we're not actually quite sure what is going on here, but I feel that this is a way for the chromatin to stay really organized and finish off the recombination pathway. And of course, when it's almost dissolved, you see that there's only little patches remaining and until there's absolutely no ASY1 remaining on the chromatin until it actually comes back. And if you were wondering if Barley was unique, well, it is in a way because the pictures are beautiful, but in fact, you can actually see such beautiful pictures as well in wheat. And this is some work that we've done with collaborators in France, in Clermont-Ferrand. And you can appreciate that the meiosis starts in a very similar way, and you have a mid-zygote in a similar way, and you have late packeting, and you also have tinsel chromosomes. But in reality, if you look very carefully, wheat also has a specific way of compacting the chromatin, and it's very actually difficult to see the three-parted structure of the synaptonemal complex. So this level of compaction, again, it's a very, very important feature. Now, something that I want to bring your attention on, it's the Meiocyte synchronicity. This is very useful because I told you that if you have 3 different anthers in one floret, they're actually around the same stage of meiosis. But the next floret will be maybe a slightly different stage of meiosis and so on. But within 3 anthers, you actually can process quite a lot of meiocytes. So you can, first of all, find a nice correlation between the size of the anthers and the meiotic stage. So this is what we've used for building up a transcriptomic analysis, which is currently under review. So the first one, first of all, is separating by size, but it's not enough. So we always do a little check on acetocarmine, which is a very, very simple stain, to give us an indication of the stage of meiosis. But even though it's not enough, especially during the prophase, early prophase stage, because this can as well be a leptotene or a zygotene stage until you really use the immunocytology to actually separate those stages, which we've done. And we've pushed this further by actually isolating the myocyte out of it. And this rotating picture is an example of what we get. So you can cut the anthers in half, remove the myocyte out of it, and transfer it in a new tube and prepare RNA's messenger for this. So it's been quite useful. And the good thing about it is it gives you a very, very pure quality myocyte and meiotic samples. So it's been very, very useful for us. So the other thing you need to know about meiosis and recombination in Barley is that you can actually plot roughly a timeline of meiosis in here on the top. And then you can plot the recombination pathway under it. So the recombination pathway is happening at the molecular level and is always start with a break. So you need to have multiple breaks all around the chromosomes in order to have your homologous recombination. A very important step is resection of this end. And the reason why it's important is because these ends will then be captured by the second machinery, RAD51-DMC1 machinery, in order to obtain strand invasion. And strand invasion is the most important things that must happen for having homologous recombination, because the DNA will be normally repaired by the sister chromosome, but it has to be repaired by the homologous chromosomes. And then you have a lot of things happening, something called the double holiday junctions, which is basically link those intermediates. You have DNA synthesis to finish the repair and move the loop across the chromosomes, and you have evolution. You can imagine that you can have a cross-shaped chromosome here for a metaphase. So it's quite controlled, and we've been quite good in looking over the years just so far. And one of the examples of the interlinking between synapsis and recombination is, for example, ZIP1 study that we've published a few years ago. So ZIP1, if you remember, you must be familiar by it now, is the magenta protein that I keep on showing you since the beginning of the talk. And if you remove it, you reduce the crossover number very, very dramatically. You almost have none to maybe a couple of crossover left, because basically the chromosomes are not close enough to actually allow for the recombination process to happen. So what we've been doing over the years is to isolate and analyze mutants. So the collection of mutants that people might be familiar about is what we call the synaptic mutant. Those were spontaneous mutations developed, found in the 70s, and they've been crossed in a common background called Bowman to create isoline, and we've been mapping those genes in those populations. And here's an example of 4 of them, which I'm going to talk and develop a little bit further in the talk. But it's only a few of them. There's less than 20 of them, so it's not very many to actually really tackle down meiosis if you consider there is about 100 genes involved in it. So we've been doing something else. We've been inducing mutations, and we've used either RNAi or we've used CRISPR. And of course, when you induce a gene, you can actually choose the one you want. So those examples are actually the genes that people have published and were probably promising in Barley. And you can appreciate that they all do something different into meiosis. So here you have like very distal chiasmata, but in this one, you actually have even chromosome breaks. So it gives you an indication that the translational research might not always be as straightforward as you thought. The other type of mutant that we've been trying to develop is what we call the T-links. So T-links basically are mutated, EMS mutated population to create mutation across the genomes. And we've identified a number of mutants based on semi-fertility because this was phenotype and normally comes after wrong meiosis. And we've tried various ways of isolating and characterizing these numbers of mutants. And one of them is actually to use cytology, and you can really appreciate that if you compare to the wild type metaphase here, they all have different conformation. So this one obviously are losing crossover, but this one also has a problem with chromatin compactions. And from that, we actually developed even an exome capture strategy to find these mutations. So it's actually quite nice because we could pool by areas of 8 by 8 and clean up the solution, the results to identify the point mutations. So we've identified 32 mutants so far from the start of 162 line from one optic populations. And we are currently trying to understand what is going on in those lines, including the validation of the mutation. So Barley is a well-behaved eukaryote and compared to Arabidopsis. So for example, if you look at packaging cells here in prophase in the Bowman genotype, and you look at the 7-ring bivalent in metaphase, and an equal distribution of chromosomes in young anaphase, this is what you expect from a normal meiosis. But of course, when you have a very classical mutant like DMC1 mutant, which is DES5, you actually have exactly the same expected phenotype what we normally have in all organisms. So whether it's mammal or Arabidopsis or any other organism, DMC1 mutant actually have an arrest in zygote. They don't proceed further. Obviously in plants, some of them proceed further because we still retain a bit of fertility, but the result is very catastrophic on the recombination because there's actually almost no recombination at all. And of course, it leads to chromosome missegregations. And the first thing we've done as well is to confirm this with genetics. Using the crosses from DES5 to BACA, for example, you can really appreciate that you have a reduced number of recombination here across the entire genome. And although most of the meiotic genes, which are very conserved, behave in the same way, we also observe some differences, and this is the story of DES10. So DES10 mutant is an MLH3 mutant, and MLH3 is a very important mismatch repair, which actually acts at the end of the recombination pathway to remove the loop that I've talked about and to resolve the crossover. And in Arabidopsis, it's been shown that you only have 60% of the recombination happening, and it's quite random. And it's also what is happening in Barley. So you also have about 60% of the recombination remaining only, and then you have only 1 in 3 crossover repaired in a random manner. But there are some differences, and one of the differences is that in your mutant, synapsis is not actually completely normal. You do have a proper alignment of the chromosomes along the entire length, but you do not have a proper polymerization of ZIP1. And it's been very difficult for us actually to count all these MLH3 foci within the cells. So this is one of the first differences that we've seen when handling a large genome for a known meiotic gene. And the story gets worse before it gets better, because here is another example of a mutant what we call Des8. And here again, I'm showing you a normal meiosis in Barley with normal leptotin zygoting and normal packaging, normal metaphase and normal anaphases. But in Des8, and in the same gene in Arabidopsis has also a normal meiosis. So this is exactly what we were expecting. So here you go. We have actually 2 alleles. The 247 allele actually is a truncated gene, the truncated version of the gene. So we might have a truncated protein or not. It might be degraded, while 248 is basically the deletion of the whole gene. So it's a knockout basically. And in fact, you don't really see much during prophase, but during metaphase, you can clearly see and appreciate that this metaphase on the top of the wild type is really different from the 2 metaphases that I'm showing you in those 2 alleles, because we are actually losing recombination. And of course, losing recombination leads to chromosome misaggregation. And what I'm saying the story is worse than that is that in Arabidopsis, this gene was supposed to increase recombination. Well, I can tell you that in Barley, it won't. It's not increasing recombination, it's actually decreasing recombination. So that's actually a bad story for us, because again, it shows you that it's not that your gene is very wonderful somewhere else, it might not work in Barley. And that's possible because of their chromosome configurations. But we have also some very, very good story coming up. So this is the story with a new gene, which we call sticky telomere 1, HVST1. And if you appreciate the diplotin stage in here with the tinsel chromosome, which I've shown earlier, and the normal metaphase and normal anaphase again, which I think you're now familiar with, here you can see that is a bit different. So first of all, this gene doesn't really form synapsis either. So we do have cell with a better synapsis, but most of the time it's not there. However, the alignment of the chromosome is correct again. So that's something which is quite interesting. Now, when we looked first at the metaphase, we were not overly excited about it, because it was not really showing us a very clear ring bivalent definition. It was actually showing some very sticky, mushy chromosomes together. And the result of it is a fusion of the telomeres here during the separation of anaphase. So we thought that possibly it will actually reduce number of recombination, or actually maybe not having any difference at all. But actually, this gene increased recombination, and is not by very, very little. It's actually increased up to 50% in some of the chromosomes. And why it's very exciting is because this gene is actually GRAS specific. So we do find some sort of orthorhog in Arabidopsis, but it's not actually even doing the same thing. It's actually the process orthorhog is involving photosynthesis. It's nothing to do with meiosis, and it's nothing to do with the functions. And we also know, and we have now the proof, that this gene is actually a novel E3 ubiquitin ligase. So this is very exciting for Barley, because it shows that there's an alternative pathway to control recombination, and that explains why we have all these differences, even if we have no gene in here. So I would like to finish with just a very quick summary. So we have a great number of resources and tools to study meiosis in Barley, and this is a very good knowledge for us, because we can actually transfer it to wheat or other crop species. And although Barley is a very well-behaved eukaryote, there are some differences when compared to the model Arabidopsis, and that's good, because they're not having the same genome, and actually give space for both races in our meiotic study. And we have found a novel gene in barley, and that is suggesting that there's an alternative pathway for control of recombination. And I can say that's probably not the only one. So I'm very excited about this research. And I would like to finish off by thanks all the people from the Meiosis group and the site, especially Robbie, for giving us the opportunity and having this nice ERC grant that we've been working on for the past few years. Past members have been very instrumental in doing some of the very ground work, like a lot of spread, a lot of CASP analysis and so on, and also all the collaborators. And thank you very much.
That's great. Thanks very much, Isabelle, for that. So again, I will encourage people to ask questions.
I can see you again.
Good, excellent.
Sorry, I've got the sun.
Yes,
sorry, I've got the sun coming up.
that must be a rare event in Dundee there, but yeah.
Well, no, we call it sunny Dundee. It's one of the driest parts of Scotland, you know that.
Apologies. So yeah, I encourage everyone to ask questions in the question box. They're really interesting talk from Isabelle. So let me start off by asking about this gene, the E3 ubiquitin ligase. Do you have any? So it sounds it's a really powerful tool that will be if you've got double amount of recombination. Do you have any idea about the mechanism, how that is working at this time?
No, not at all. And that's the problem, because it's novel. It's not like all the other ligases or other genes that have been described already. You know, I mean, it's a lot of genes have been described in Arabidopsis or in human or mammals, but this one, I can't find anything anywhere. And there's no a closest ortholog. You can always find maybe 20% similarity with a protein, but it doesn't actually mean anything. So this is why actually I'm quite happy that I have a grant starting, because the grant is actually to explore this a lot. And we're going to do it by various ways. So we're going to do it by pooled assay, because we actually have made an antibody against this protein, and it works quite well on Western blotting. And then we're going to do as well a ubiquitome using the diagonal assay, and we actually have a collaboration with University of Dundee to try to do this work, because we have no idea what it does. And myself, I was actually surprised, because when we looked at the metaphase and when we looked at the synaptonemal complex formation, I really thought that this gene was actually acting early on in meiosis, but in fact it's highly expressed later on at packaging. So that was another thing. And the other surprising things, I thought that it could have been high 10 initially, which is high 10 is another E3 ligase that's come up increasing recombination, but it's not. It's actually not even acting in the same time at all. So it's very, very exciting, because it's different, and it's very specific to barley, and I know there's one in wheat because I've checked. It's class specific. You can see it in barley, you can have it in rice, and you can have it in sorghum, in rye, but Arabidopsis is very, very far away, so you can't really say that is the gene. So I think it's very exciting to see that there's a difference in the way it's controlled recombination. It makes sense now, actually.
Absolutely, it'll be fascinating to find out what the targets are for that ligase. So I have a question from Andrew Lloyd, who asks simply what's the Arabidopsis homologue of the DES8 mutant?
I can tell you, because the paper is almost ready. It's XRCC2.
Okay. And okay, and so and much known about that protein in Arabidopsis?
There's quite a lot of things that known, and it's fascinating, because it's increasing recombinations, and the meiosis is normal. It's acting in the RAD51 and DMC1 pathway, and I can tell you that there is something happening with RAD51 and DMC1 in barley, which is a little bit different. So I mean, we're really preparing the manuscript just now, so hopefully we will, publish it, at least submitting soon. Hopefully.
Okay, that's good.
But
I mean, it's super interesting that you're finding that there's all these quirks and differences in barley, which are giving you a real real reason to study barley, because obviously then, when you look to the breeders and that, there's going to be these all specific things for barley. That's fantastic. So I have a question from Pierre Deveau, and he asks, in non-crossover events, only a small piece of DNA, 20 base pairs of homologous chromosomes is exchanged. Does this occur in barley as well? And do you know why there is? I don't know.
I'm not sure about the lengths and everything. This is, there's a lot of things to look at. So we are not, at least in our group, we are not in these molecular levels. This is something that I would really like to explore. I think the non-crossover pathway have never been very explored just now. This is a question we've had since the DES10 publication, because the theory of an MLH3 mutant is that MLH3 increase the number of non-crossover, and we don't have any way of measuring it just now. So yeah, I'll be interested to see if anybody has any idea how to make it happening.
Okay.
Yeah.
Okay, let me just finish off then by asking, so obviously you're going to be looking at this new work with the E3 ligase. You're going to be looking at the nuts and bolts, how it's regulated there. But let me ask you about the pathway. So if you find some interesting things that could be relevant for breeding, how do you bring that into the into breeders and things? What sort of relationships have you got, the James Hutton, to those guys?
So I think well, okay. So for us, we have something called the International Barley Hub, actually trying to expand basically, and it's based on a very strong collaboration we have with the James Hutton already and breeders. So we already have contact with breeders and things, and I think what we need to do, and the only problem, ST1 is a good one because it's a spontaneous mutation, so there's no issue with transgenic and stuff, and that's the problem that stops some of the breeders, because all the other gene we had to make induced mutation. This is why we've made them. So genes such as usual suspect, like FANChem, for example, we've made transgenic, but the problem is the breeder will be a bit reluctant to use them because they are transgenic, right, in the breeding process, rather than ST1 is actually very good because as a spontaneous mutation, we can use it. But there's a drawback to it because the problem of these lines is they're semi-fertile. So there's a cost of fertility that is very difficult to work with. And it's quite severely fertile, even though, because of the problem that you get at Synapsis. So I think it will be worth to explore to see in crosses if you actually release some genetic diversity. And there's a nice work that's been done by one of our students, Mikel Ayeta, who actually looked at the cold stress in plant, and the cold stress actually increased the recombination in the telomeres, and they increase a little bit the diversity here. So I think, to me, depending on where your gene is located, you can actually use those tools to accelerate and maybe cut your breeding. Instead of having a crop and a variety in 10 years, maybe you'll have it in 5 years. And I think for a breeder, it's very, very important. But one of the most important things, one of the most important things as well is to actually release the genes which are trapped, because the problem of these genes, they can actually be very important. They can reduce fertilizer, they can reduce, pest and disease resistance, and we can actually increase pest resistance without having to use fungicide or something like that. So
so maybe a final question then, just linked to that idea about the environmental impact and the cold stress you mentioned. So Zoe Wilson asks, are there any environmental impacts, such as temperature, which are associated with the ST1 phenotype?
So with ST1 is not particularly I'm not entirely sure. We've done I think it's the one actually was not on the panel of the heat stress for some reason. But I can tell you XRCC2 is a bit more resilient. For example, DES8, if you put in heat stress, it has actually changed very, very much. So it gives a bit more resistance. You think that it will increase and worsen the fertility, but actually it's not. So depending on the mutant, and in fact, the DES5 mutant I talked about is actually even better because it's actually increased fertility. So if you put in heat stress, it's actually restoring the fertility, and I think it's linked with the DNA replication timing and the way it's organized. So I think every single one of them, depending on where it's targeted in pathway, will have a different impact. But unfortunately, ST1 is, I think, the only one we didn't put very enough on the panel, and I think it's because we didn't have enough seeds to do the experiment we wanted to do, and this one at the same time. So that's something we're going to look at.
Absolutely. Okay, so let's leave it there. So thanks very much, Isabelle. And